Welcome to another episode of the Common Babbler podcast, where we deep dive into a series of conversations that discusses science pedagogy with a focus on how effectively we communicate science in an undergraduate classroom. I'm Kalyan Chakrabarti. I'm an associate professor of biological science and chemistry at the School of Interwoven Arts and Sciences, Krea University. And we have our guest today, Professor Kevin Gardner. Professor Kevin Gardner is the director of the Structural Biology Initiative in the City University of New York Advanced Science Research Center and Einstein Professor of Chemistry and Biochemistry, City College of New York. So, Professor Kevin Gardner received his BS in biochemistry from UC Davis and then obtained his PhD in molecular biophysics from Yale University. And then after his postdoctoral work, which has been very fundamental in the field of protein spectroscopy, protein nuclear magnetic resonance spectroscopy. And as a student of that method, we all had to use his breakthrough during his postdoc. Then he started his structural biology research group within the departments of biophysics and biochemistry at the University of Texas Southwestern Medical Center in Dallas. Professor Gardner now serves as the inaugural director of the Structural Biology Initiative of City University of New York Advanced Research Center and as the Einstein Professor of Chemistry and Biochemistry at the City College. He leads in the Advanced Science Research Center. It's a new interdisciplinary research enterprise built by City University of New York. Welcome, Professor Gardner.
Thank you very much. I'm very glad to be here.
Thank you for being with us today. And I think, so I should mention here that this effort is independent of our affiliations and responsibilities, including in teaching and research. I wanted to ask you first that, and I think I left out in the introduction that you are also a certified scuba diver.
That is correct. I am. And I've been here in India providing a series of lectures up in the north a bit and now later today. But I've also been a student, taking advantage of some time down in Pondicherry to get some of my expertise tuned up in certain aspects of scuba diving, which included some practical public science yesterday of going to 25 meters down, cracking a chicken egg open, and watching the effects of pressure on it. It's absolutely beautiful. The chicken egg yolk is this beautiful yellow sphere. It's almost perfect, and you can move it around like a ball in between various things while you keep it intact.
So does it immediately become solid when it's cracked open?
No, it's not yet a solid. I would describe it more like a soft liquid. So you can see it as it is moving around. There are ripples occurring on the surface. But instead of being flat on a plate, as we might see as we're cooking it or preparing it in the kitchen, it's actually this beautiful yellow sphere. It's just like the sun. It's absolutely beautiful. And the whites, of course, fritter off and dispel almost immediately, leaving just this beautiful yolk. Absolutely fantastic.
It's fascinating. How dark does it get at when you are? So you said that you went to 25 meters and then and the deepest was 37.
37 meters on a later dive, absolutely. And it's great there again being a student. I was saying earlier that makes me feel young again to go in and have to learn some things that are actually the bulk of them are, quite frankly, the kinds of things we learn in general chemistry.
Right.
We have a tank on our back of a fixed volume of gas. We're changing pressure by whether you're at the surface with 1 atmosphere of pressure or every 10 meters down the water at another atmosphere of pressure. That's in the classroom is kind of a trivial thing, but when it determines how long you can stay underwater before you run out of air, it becomes kind of a bit more important to know your general chemistry.
Fascinating. You had very interesting work, which I have been following for quite some time, in which these proteins, they are called the LOV sensors. I think light, oxygen, voltage.
Correct.
And these proteins are, if I understood correctly, these proteins are involved in, they are present in plants and they kind of sense the light to, so that the leaves change their physiological state.
Right. So this is something that farmers have known about for literally for thousands of years to go through and understand how plants sense the daily dark night cycle. A very good friend of mine, the late Winslow Briggs at Stanford University, was one of the first to really go through and take a look at a molecular basis how this worked, how photochemistry, a photochemical change of a structure inside of a protein, could trigger a set of reactions leading to, like you said, leaves trying to be, for example, as open as possible in dim light to capture any photon they can, and then at nighttime to help reset the plant for the next day. It turns out that the law of photochemistry, however, is much, much, much more general. Literally, it's been found from those initial handful of proteins that Briggs and coworkers identified in literally thousands of others that synchronize the circadian clock of molds, of helping tune responses in marine bacteria, for example, like in the waters I was diving in. For them, knowing whether there's enough light or not, and for the law of domains, it's blue light, is a sign not only of whether it's day or night, but whether you're swimming on the surface of the ocean or deep down inside the ocean. Again, things that are fundamental for them to make decisions of where do I get my energy? How should I move? Should I make copies of myself or not? Very fundamental.
So recently you have published a work, and this is this fascinating tool in biology, which is the optogenetic tools.
Absolutely.
And you have used that concept, if I understood correctly, to convert it to a tool in order to illuminate the cells under microscope.
Absolutely. So this is a field that is this optogenetics, this repurposing of genetically encoded photosensors. And again, nature has been working on this problem for millions of years. How most effectively to detect light anywhere across the visible and even sometimes out of the visible spectrum. For example, plants and bees can detect in the ultraviolet. Plants are very good at doing some interesting mathematics in the infrared, as it turns out. So taking the sensor proteins that handle that and converting them for new purposes, new technologies that hadn't been anticipated before. For example, giving a cancer researcher the chance to turn on or off a signaling pathway in a set of cells in a tumor simply on part of a tumor and not on the rest of a tumor, or maybe going into, say, a genetically engineered mouse, having two tumors inside of it, illuminating one to test a new therapy idea while leaving the other unaffected as a control. Extraordinarily powerful. It's been transformational. That's not a word I use lightly. It has literally been transformational in the field of neuroscience, where a number of coworkers in the field, Peter Hegeman, Carl Dijsselroth, Ed Boyden, and others, have gone through and engineered light-activated ion channels to control signaling through nerves with light. Now it has expanded into many other kinds of activities that can be controlled. Even, for example, in our work, turning genes on and off, using, for example, a sensor that initially evolved in a marine bacterium to let this very simple organism now know whether it's day or night, whether it's on the surface or deep in the water. It's now in hundreds of labs, turning genes on and off that have absolutely nothing to do with its original purpose, but ones where researchers have come up with very creative ways to take advantage of the fact that with light, like you mentioned, you can go under a microscope or into a whole organism, only illuminate a certain portion of your sample, and you can supply that light for fixed periods of time. Then when you're ready, take the light away and shut the stimulus off immediately. Those are things that are almost impossible to do with any of the conventional methods we currently have in the area.
So that's about the tool. And also, this is a fascinating thing, I think, where the nature is, in a way, uh, uses the same fold or the same design in multiple places. And also, speaking of cancer, I think your recent work is on the hypoxia inducing factor, that HIF. And you have shown that it has the same fold that is found in the LOV sensors in plants.
Absolutely correct. It's the same fold, but then a slightly different pocket or cavity within the protein, which in the LOV domain holds a chromophore, a natural molecule that's very sensitive to light. It's actually one of the standard vitamins we should all be eating in our diet, called flavin mononucleotide or riboflavin. You may have heard of it more commonly. Over in the HIF, we actually don't have a great idea as to what the natural ligand is, but we do know that we've been able to get artificial compounds to head into a very analogous site to turn that system on and off, now not with light, but whether we supply this compound or not as a reagent in a test tube, but really more critically as a drug in the clinic. So for your listeners, the HIF pathway is one that is used for oxygen sensing in our bodies. It is critical when your cells are starved for oxygen. They need to be able to respond to that to adapt and recruit more oxygen carrying blood vessels, for example, into the area. However, when the system's turned on all the time, you get a very high probability of developing cancer quite early in your life. As a result, there's been intense interest in developing novel small molecules, novel drugs to be able to turn this system on and off. And I'm intensely proud of work by my research laboratory and that of our collaborators, both in the academic and industrial sector, that have been able to turn this from being a crazy idea into something that is literally addressing cancer in thousands of patients currently in the US and UK.
Fascinating. You have worked on the UT Southwestern, which is a medical school,
Mm-hmm.
and now you are in the City University of New York, which has many other disciplines, and it's the atmosphere changes.
Absolutely.
And this is a debate that we hear often, that is there any simple answer to which one is works better or which one is.
Oh, I think that's a false question, with all due respect, because I think the answer, which one works better, I think fundamentally gets to the heart of what is the purpose of education and who is participating in the exercise. UT Southwestern is, as you noted, is a very focused organization. Roughly 2,000 students there at any given time, 1,000 of whom are proceeding towards a medical degree, postgraduate degree in the United States. The other 1,000 are pursuing either Ph.D.s for the sciences or a variety of degrees in the so-called allied health, nursing, physician's assistant, other endpoints from there. At City College of New York, the flagship of the CUNY system, we have 12,000 students proceeding in undergraduate degrees across, like you said, an extremely wide spectrum.
Mhm.
Fiercely proud of the fact that I think we have about 100 native languages, the attendees who are there. Many are first generation going to university, many are first generation in the United States, many are both. And the one thing that I think I was warned about is something that I challenged upon arrival was a variety of voices saying the ideas at the medical school could not translate to the university environment. And that I challenge. And I'm fiercely proud that, for example, in my undergraduate biochemistry classes, which are for year 3 university students, the things we're teaching are actually things that are quite common, I think, for many of their peers to only get introduced to for the very first time in the first year of medical school, which would traditionally be 2 years later for them. And it is not only science, what kind of science they're learning about, but a bit of go beyond what's in the textbook. What are the things that are breaking? What are new? Number one. And number two is really engaging the students in learning the absolutely critical part of knowing how to communicate their work. And while some of them may find the assignments onerous, to go through and go pull something out of the scientific literature, which is very technical writing, very sophisticated ideas. Sometimes I think you learn best when you are thrown into an uncomfortable situation and you need to adapt and learn from it. And then learn how to communicate what you've learned to your fellow students by, for example, writing a 5-page paper explaining those complex ideas in terms that their peers can understand and appreciate, or stand up in front of class of 80 of their peers and give a 90-second, one-slide PowerPoint. What did I learn? Why is it important? And why does it have anything to do with what we're learning in class? Those are ideas that I imported after really test driving them, if you will, up at a PhD and medical school level for quite some period of time before going to the university level. And I think it's that kind of innovation I think my students have appreciated. I hope any who are listening to this podcast would agree with that. But it has really been fun from my standpoint as an educator to be able to go put into play.
So another thing that comes up in debates frequently, and especially in a university which has education and which has large undergraduate class, is that, for example, the science, natural science, especially biology, is expensive. It takes resources, biological research. So I think it's kind of a two-part question. So one is that how critical is that research component for teaching, and especially for an undergraduate program? And in general, since the society is spending quite a bit of uh, resources on the biological science research, let's say, the basic research, uh, what can it expect in return? Or how do you see
Yeah.
it? How do you sort of justify it?
I wish we had another hour to
Yes.
discuss these topics because these are very fundamental to who I am, both professionally and personally, to be blunt. So, to the first question, is research essential? Yes, it is essential for the teaching mission, in my opinion, for researchers to be teaching and teachers to be researching. Again, one thing that I had been told coming into CUNY by some is that it was impossible for a researcher to be a good teacher and vice versa. With all due respect, I challenge the assertion and would insist actually that I think, while it is not simple, I wholeheartedly agree for reasons of expense, for reasons of commitment of time, for reasons of infrastructure, to be set up to do both. Doing both is absolutely critical for giving students an education and introduction to the latest groundbreaking aspects of science. And then I think quite critically, as a student myself who I found theory discussions in science too abstract, to be blunt. I could go do them if I had good inspiration to, and one of the best inspirations was by going into the lab or knowing I was going back into the lab to go do some of the complex nuclear magnetic resonance experiments, for example, that you alluded to before. Having that practical insight was a great motivator for me to go in and learn the theory and really make those lessons much deeper. Your second point on the investment in research and this critical issue in the US and in Europe, I know for certain, as I imagine here, it really oftentimes gets broken into a discussion of basic research, fundamental research, and translational research. I hope the vignette we spoke of a few minutes ago is a great example of exactly why those things need to happen hand in hand. And I think we could go where blue light sensing on one side of my lab, but again, not the photoreceptors in our eyes, but instead things that are happening in plants or obscure marine bacteria. Having that go on side by side with people interested in oxygen sensing in cancer, absolutely essential to have those going side by side. As we look at now, the revolutions we're seeing over in aspects of genetic engineering and aspects of vaccine design, most recently, for example, with the mRNA vaccines that are popular in the United States. I don't believe they've reached to quite the same level here in India. But the fundamental science for that happened years ago and for a very different purpose. CRISPR, genetic engineering, same story. Understanding how bacteria fight off foreign DNA has become integral to aspects of genetic engineering that I think will be absolutely transformational for agriculture, for medicine, and potentially for a variety of societal things that we all need to be thinking about. None of those would have happened if one had solely focused on the short term. I have a disease. How do I treat that most immediately today? Don't get me wrong, that's still critical. But the two of those are integrated in far more ways than I think any of us can really comprehend.
Thank you so much, Kevin.
Thank you.
Thanks for your time, and really look forward to many more interesting tools coming out or the applied research coming out of your lab.
Rooted in fundamental research. Yes,
Yes, rooted in
absolutely.
fundamental research. Thank you.
Thank you very much.